Friends, today we are going to discuss about the structures of the DNA. Most of the peoples are very well known about the major functions of the DNA. So today we are not going to discuss the functions of the DNA. We are going to concentrate only the different component of the DNA as well as the structural features of the DNA. So, in 1953, two scientists, James Watson and Francis Crick, has proposed the 3D model of the DNA. Based on the previous scientific information generated by the various scientists, uh, they have proposed this structure. So they have the various scientific informations like the X-ray diffractions image taken by the Rosalind Franklin and the Raymond Goslings, as well as they have another informations like the base pairing of the DNA, as well as they have the structural informations of the nucleotide. So based on that previous informations, they have suggested the 3D model of the DNA in 1953. So DNA, it is a made up of the nucleotide. It is a polymer of the nucleotide. So nucleotide structure, it is a made up of three major components. One is the nitrogenous base, another is the pentose sugars and phosphate group. So nitrogen base are two types. One is the purine, it is a double ring structure. Another is the pyrimidine, it is a single ring structure. Purine includes the adenine and guanine, and pyrimidines include the cytosines and thymine. So in a DNA, A stands for the adenine, G stands for the guanine, C stands for the cytosines, and T stands for the thymine. Nitrogenous base are attached on the pentose sugars, that is the deoxyribose sugars, on the first positions of the pentose carbon or deoxyribose sugars carbon first. So deoxyribose sugars means that there are one oxygen less as compared to the ribose sugars in the second carbon positions, and phosphate groups are attached to the deoxyribose sugars at the 5' end. So this is the structures of the nucleotide. So in a nucleotides, there are at the third positions one OH groups are there, and this is remain free. So during the polymerizations, the successive nucleotides are attached at this third position. So phosphate groups are joined at the these carbons atoms or this third number of the carbons atoms where the OH groups are remain free. So there are a successive every nucleotides are joined in a third carbon's position. So there are a it becomes a phosphodiester linkage. So every nucleotide makes a phosphodiester bonds with the successive nucleotides. In a DNA strands, these are known as sugar phosphate backbone. So these are the major features of the polymer of the DNA. So according to the 3D model suggested by the Watson and Crick, it is a double helical structure. So DNA, it is a double helical structure, and it is a right-handed helix. So due to the specific turns, it forms the major grooves as well as the minor grooves and their structures during the helix. Moreover that both the strands, it contains the sugar phosphate bonds are remains outside and the nitrogenous base remains the inside. So these are the features of the double helical structures. So it contains major grooves, minor grooves, and nitrogenous base which are remain inside, whereas the sugar phosphate backbones are remains outside. So this is the sugar phosphate backbones and it remains outside, and nitrogenous base remains inside the double helix. It is a anti-parallel orientations. So one strands are running in 5' to 3' directions in this way. Its opposite strands or its complementary strands are runs from this direction to this direction from 5' end to the 3' end. So this is anti-parallel in nature. So if here it is a 5' and its opposite directions, always there are a 3' end, and if here it is a 3' and opposite direction, it is a always 5' end. So this is the anti-parallel regarding the anti-parallel orientations. Another main features of the double helical model of the DNA, it is a complementary base pairing. In a complementary base pairing, if there is a adenine in a one strands, in a opposite strands it contains always thymine. Whereas in case of the in a one strand, if there are a cytosines, in a opposite strands there are always a guanine. So likewise that A opposite there are a T and C opposite there are a G. And if here if it is a G in a opposite direction, it is always C, and if here it is thymine in a opposite directions, it is a thymine. It is because during the DNA replications, the one strands it is a act as a template strand, and the newly synthesized strands it is always add the nucleotides which is complementary to the template strand. So daughter strands contains the uh, complementary base pair in their structure. Moreover that these both the strands or both the helix are stabilized with the hydrogens bondings between the two nucleotides of the opposite strand. It is due to the nitrogenous base. It's shares the different type of the hydrogens bonding. So between A and T there are two hydrogens bonds are there, and between G and C there are three hydrogens bondings are there. So both the double helix are complementary to each other and they are stabilized with the hydrogens bondings. Moreover that uh, it contains a per turn almost 10 nucleotides, and per turns there are a distance per turns it is a almost 34 angstroms. So between two nucleotides there are almost 3.4 angstroms. It is a distance between two nucleotides. So this is the other major features of the double helical model of the DNA. And this distance, so between these two strands, there are a distance is almost 20 angstrom. In general, there are a different kind of the DNA present in a natures like the aDNA, bDNA, and zDNA. So these are per turn nucleotides. It is a differed according to the type of the DNA. But in general, the distance between two strands it is a 20 angstroms, and 10 nucleotides it is a 34 angstroms in general. So friends, if you like this video, please press the subscribe buttons, and thank you for watching.